Hello everyone, welcome to Ultrazone Plus, and today's topic is the staphylococci. And the word staphyl is derived from the Greek word which means bunch of the grapes, and there are 40 known species of the staphylococci. And with the gram staining technique, they give gram-positive reactions, and they ranges from zero point 0.5 micrometer to 1.5 micrometer, and usually in the form of clusters, pairs, tetrad, or short chain. And on the basis of these arrangements, there are different kinds of these bacteria, and there is absence of the flagella and spores, and they form capsules which are variable, like they form pseudocapsules or some other type of the capsules. These are facultative anaerobes and require simple media for their growth, like 37 degrees Celsius temperature required for their optimum growth. And there are different kinds of the media for these bacteria, like nutrient agar, blood agar, egg yolk medium, McConaughey agar, liquid medium. So on the nutrient agar, they form colonies which are 2 to 4 mm, which are circular, convex, smoothie, shiny, and the pigmented. On the blood agar, they show beta hemolysis, and on the egg yolk medium, opacity on the glucose egg yolk medium, and on the McConaughey agar, they form small and pink color colonies. The liquid medium form uniform turbidity, and the selective media and the most appropriate media for these bacteria with the 8 to 10% NaCl. All the Staphylococcus species except the Staphylococcus sacrolyticus and the Staphylococcus aureus subspecies are the facultative anaerobes. They grow best in the presence of 10% NaCl and produces white, yellow, or golden yellow colonies. And they are generally catalase positive and produces coagulase, which increases their pathogenicity, and they ferment sugars like glucose, lactose, mannitol with the acids and produces pus in the lesions and liquefy gelatins. These bacteria show resistance to the heat and dehydration and mostly killed at 60 degrees Celsius in 30 minutes. And they are also killed by the disinfectants like crystal violets and survive in the tissue and the pus for the long time. These bacteria are classified on the basis of pigment production and the pathogens. On the basis of pigment production, the Staphylococcus aureus produces golden yellow colonies, the Staphylococcus albus produces white colonies, while the lemon yellow colonies are produced by the Staphylococcus citrus. And the pathogenicity, these bacteria may be of pathogenic or the non-pathogenic. The pathogenic species include the Staphylococcus aureus, while the non-pathogenic species include the Staphylococcus epidermidis. This pathogenic species, also known as the Staphylococcus pyrogens, these pyrogens affects on the hypothalamus and increases the temperature of the body. Mostly these are opportunists, mean they need some opportunity to show their pathogenicity, and they lives on the skin and the mucous membrane and break sometimes the epithelial barrier to allow them to enter into the host and become the pathogenic. Staphylococcus aureus. The Staphylococcus aureus is the most important species, and a variety of diseases caused by these bacteria and contains the virulence factors like some coagulases and different other to show their pathogenesis. And after 35% of the humans persistent nasal carrier, and these are easily transferable from humans to human while skin contact, and the important in the infection control, especially in methicillin resistant Staphylococcus aureus or the MRSA. Staphylococcus aureus causes the nosocomial pneumonia and skin and soft tissue infections, and the second most common cause of the primary bacteremia. And the colonies formed by these bacteria are pigmented, which are in the form of cream yellow to orange, and the cell wall is pigmented, which are carotenoid and show hemolytic on the blood agar. There are some biochemical characteristics of these bacteria, like these are catalase positive, coagulase positive, and show the slight agglutination. There are some localized infection caused by these bacteria. You can see in the pictures, these are impetigo, boils, and the carbuncles. Very common cause of infection by the staphylococcal species, often result in pus formation and can result in skin, soft tissue infection, or the deep abscesses. Any localized infection may become invasive and lead to deep bacteremia. And there are two kinds of infection, like systemic infections and some of the toxic mediated diseases. The systemic infections include the primary bacteremia or secondary bacteremia, and the endocarditis means inflammation of the endocardium, and the meningitis or the inflammation of the meninges. And some of the toxic mediated diseases, these are staphylococcal toxic shock syndromes, staphylococcal food poisoning, staphylococcal scale skin syndromes. So these are some oral infections or the systemic infections. So these are acute parotitis, angular cheilitis, and mucositis. And there are some toxin mediated like a toxic shock syndrome, toxins are TSST1. These include the superantigens and capable of activating large number of the T cells and perform heat resistant enterotoxins like staphylococcal food poisoning, and these symptoms occur in 2 to 6 hours, and these are self-limiting, depends from the person to the person. And the next one is the MRSA, methicillin resistant Staphylococcus aureus. These show resistance to the penicillin, cephalosporin, and the pennins, and usually show multiple resistance to the multiple drugs, while there is low chance of the resistance to the vancomycin. Acquired hospitals and the community acquired methicillin resistant Staphylococcus aureus. There are some exfoliative toxins A and B, which causes scale skin syndrome in infants or neonates. Then the PVL, which contains the F and S components and causes the lysis of the white blood cell and led to the massive release of the inflammatory mediator responses for the necrosis of the cell death and severe inflammation too. And PVL is the also important virulence factor in the methicillin-resistant Staphylococcus aureus infections. And the treatment includes the drain pus and the foreign bodies are removed from the dead tissues and methicillin, erythromycin, clindamycin, vancomycin, and topical agents like mupirocin. I hope you liked the video. If you like the video, please subscribe the channel and click on the bell icon for the new video. And thanks for watching.